Hello, everyone, and welcome to our second seminar of 2024. Today, we have a great talk coming up for all of you guys today. But before that, just a quick reminder that you can ask your questions in the YouTube chat at any point during the seminar, and I will ask them to our speaker after the talk. So, today for our talk, we would like to welcome Dr. Beth Nicholls as our second speaker of 2024. Dr. Nicholls completed her PhD at the University of Exeter in 2012. Since then, she has held several postdoctoral and visiting researcher positions in the United Kingdom, Germany, and India. She also founded her own interdisciplinary research lab in 2020 at the University of Sussex. Her lab's research focuses on nutritional ecology and plant insect interactions in various bee species. Outside the lab, Dr. Nicholls and other members of her lab actively engage in various outreach activities focusing on bees and pollination. Dr. Nicholls was also awarded the 2022 Crotto Award for Public Engagement in Life Sciences for her outstanding commitment to outreach and raising awareness for bee conservation. You might also have found her featured in various media coverage outlets, including BBC, the New York Times, and the Planet PhD podcast. Today, she will give a talk entitled Behaviour to Save the Bees. And with that, I would like to introduce and welcome Dr. Beth Nicholls to our talk. Welcome, Dr. Nicholls.
Thanks very much, Brian. And thank you very much to the Animal Behaviour Live Committee for inviting me to give this seminar. I think it's a great organisation and initiative, and so I'm really happy to be here speaking to you today. This talk is going to be a little bit autobiographical, actually, and sort of take you through my research career a little bit, from starting with really kind of blue skies, basic research questions in animal behaviour, to then incorporating some more applied and conservation relevant questions into my research and how I kind of got there. So I'll talk a bit about some research I've done along the way, and then finish up by telling you a little bit about what we're doing in our lab at the moment. We don't have lots of data to present yet, but we are all frantically kind of trying to analyse our data so that we have things to present at conferences this summer. So I'll also flag where the research is going to be presented so that you can check it out in the future as well. So, as Brian mentioned, my research career started way back in 2007 at the University of Exeter in the southwest of the UK. And here I studied for both a master's and a PhD in animal behaviour. And all I kind of knew at that point was that I was interested in behaviour, and I really thought insects had some of the most interesting kinds of weird and wonderful behaviours. And so I ended up doing a PhD on honeybees predominantly, and looking at kind of these basic questions around their sensory ecology, their cognitive abilities, how they decided which individuals would forage for pollen or for nectar, because in honeybees they tend to specialise in one behaviour or another, and kind of what are they learning about these rewards when they visit flowers. But around the same time as I started my PhD, kind of there became increasing awareness about bee declines or insect declines more generally. But actually, what kind of really brought this at that time to kind of the forefront of the public and the media's attention was that bee farmers in the US started to notice that their colonies were really declining. Bees were completely absconding from the hive, so they'd come back to their colonies and find no bees in there. And in the US, bee farming is a big industry. Some of you might already be aware, but the colonies get sort of shipped around from state to state following the blooming of the different crops. It's the same colonies that get moved around. Successful for them, but these absconding events were becoming really marked and really noticeable, and they coined this term of colony collapse disorder. And so this really brought this decline to the attention, as I say, of the media. But probably insect declines had been going on for a lot longer than this. It's just that maybe people don't care so much if a fly or a midge or a little beetle is disappearing. But bees as a kind of, or honeybees as a kind of managed pollinator of commercial importance, people are going to start to notice, and they're going to start to talk about it. And so this led to a lot of media coverage, a lot of public interest. People really like bees. I think it's because they're fluffy, to be honest. They have quite big eyes. They're pretty charismatic. So they've become this kind of poster child for insect declines. And so, as I say, this sort of started to happen just around the time I started my PhD. And anyone who is currently studying for a PhD or has studied from a PhD will maybe be familiar with these questions from well-meaning family and friends who are wondering what exactly it is that you're doing with your time. Why are you still at school in your 30s? They don't really necessarily always understand what we do. And so for me, this was really compounded by the media talking about bee declines a lot, people becoming very aware of it. And so my friends and family were asking me, oh, what is your PhD doing to help save the bees? And I was thinking, not a lot, really. I'm probably, euthanizing quite a lot of bees as part of my project. So it did kind of get to me a little bit, and it did make me start to think. And for me personally, I was feeling a little bit demotivated by the end of my PhD. I think that's not an uncommon experience either. You get a bit bogged down in it. You've been working on this one thing for so long. And I did start to realize that I think the reason I became interested in animal behavior was because I was interested in the natural world and in conservation. And that maybe for me, having some kind of applied aspect, some conservation aspect to my research was quite important to help me kind of keep motivated. And so I think Blue Skies research is incredibly crucially important. But for me personally, I did start to realize that, having some applied aspect would be beneficial to me personally as well. And of course, if you work with pollinating insects, that's not too hard to find that kind of applied angle. So I made a really conscious choice to start looking for positions where I could use my training and background and knowledge of studying behaviour to try to answer some of these more applied questions and to try to start to understand what might be driving some of these declines in bees, but in pollinating insects more generally. So before I start to talk about some of the research that I was involved in, I thought I should really get the doom and gloom out of the way, give some evidence for these insect declines, and also talk a little bit about what might be driving them. And then I will talk more about kind of the research that I've been involved in and how I brought behaviour into it. So going back to this image that I showed, I mean, it really does nicely show that honeybee declines or the declines in honey producing colonies in the US are quite significant. There's a point in time where Varroa, this mite that affects honeybee colonies, became introduced into the US, and that was thought to be one of the drivers. But honeybees are, as I mentioned, a managed pollinator. So that means that they have a beekeeper for the most part, and the number of honeybee colonies, to a large extent, does depend on how many people are keeping bees, how economically profitable it is to keep bees or to be a bee farmer. And so it's often suggested that, using honeybees, although they might suffer from some of the same stressors as our wild bees, but using them as a kind of barometer of bee declines, or as a barometer of the numbers of bees we have, is sort of akin to kind of counting chickens, another kind of domesticated animal, to try and understand bird declines. They're not under the exact same pressure, and they're not, for the most part, wild animals. And actually, kind of to demonstrate that, although there were declines in the US and in Europe over this time period, actually global stocks of honeybees had been increasing. And that's due to increased interest in keeping European honeybees in South America and in Asia. So the honeybee picture kind of clouds things a little bit. But the problem is that for our wild bees, we don't have great data on abundance. So there aren't very many, or there definitely haven't been historically very many kind of formal monitoring schemes where we are counting how many individuals we have of the various different species. But what we do have is distribution maps. And I think it's worth flagging that often this data of these distributions comes not from scientists or from ecologists, but from very interested and passionate members of the public who just have a passion for insects and go out in their spare time and record these observations. And without them, we wouldn't have these long-term kind of distribution data sets. But the picture here is pretty depressing for many species. So this is the distribution change for the great yellow bumblebee. I've never seen this species. It used to be quite well distributed across the UK, but now you can see it's really restricted to the Highlands and Islands up in Scotland. And we see similar range contractions for species in the US, such as the rusty patch bumblebee, which actually was one of the first insects, I believe, in the US to kind of get this actual conservation status. And hopefully things are being done to try and preserve this species. For all the other bees, we have even less good data. Many bee species are very cryptic, so it's quite hard to distinguish them on the wing. You need to do lethal sampling, which not everyone's up for. But we do know that in the UK, at least, we've had 23 species go extinct over the past 150 years. Data for butterflies is a little bit better, and we do have better abundance data for these species. They have been monitored for longer and more formally. They're, at least in the UK, we don't have that many species, so they're a bit easier to distinguish on the wing as well. But again, the stats are pretty depressing. So we see massive declines, particularly in species associated with farmland, which might tell us a little bit about what some of the drivers could be. But if you were really cynical and you only really cared about bees or pollinators because of the services they provide to us, because they help pollinate a lot of the food that we eat and depend on, then you might take the approach that, well, as long as honeybee stocks are going up around the world, then why should we care? because we will have enough pollinators to pollinate the food that we eat. Hopefully no one thinks like that in this audience, but you never know. But of course, flowers and plants have a huge diversity in floral forms. And this is in part due to co-evolution with those insect pollinators that provide that key part of their reproduction cycle to them. And many plants will try to restrict a little bit which insects can visit so that they're not getting too much heterospecific pollen deposited, so that they're getting very efficient and accurate pollination. And so they have all kinds of innovations that require specific behavioral routines, such as solanum species that need to be sonicated to release their pollen, or keel flowers that require the bees to kind of push down a petal to access the nectar rewards, or other adaptations like the foxgloves here in the bottom left that have hairs that restrict the access of the pollinator. So not all pollinators are kind of able to adequately pollinate all plants. I guess many of you already know that, but I think it's worth reiterating. And things like their morphology, such as their tongue length, but also kind of the behavioral routines that they engage in, will affect kind of how effective they might be as pollinators of these crops. So whether they actively collect pollen from the flower, whether they pack it away in leg sacs where it gets mixed with nectar like the honeybee in the top picture and cannot be used in pollination, or whether they simply kind of get dusted with the pollen as a function of visiting the flower for another reason, will really affect how effective they are at pollinating a particular flower. And things like body size as well are really important. So the rose chafer beetle in the top right picture will not be able to visit flowers with very small openings, for example. So something like the squash flower is going to be visited by those smaller squash bees or the honeybees. So there is increasing kind of empirical evidence as well to show that just relying on one species of bee for our pollination needs for our food crops is not going to be particularly sustainable, and actually we get much better fruit set and higher yields where there is a diversity of insects present. And hopefully you can see from this figure that for some crops like the tomatoes, which require buzzing, or things like coffee, which are pollinated by a really, really tiny midge, cannot be pollinated by honeybees. So preserving that diversity and understanding more about those other insects that are providing pollination services is important, I would say. So in terms of the causes of pollinator decline, I was fortunate enough to be involved in a review led by Dave Goulson that kind of reviewed these drivers, which I now realise is nearly 10 years old, so probably need to update, but I'm sure some of these stressors are still having a big impact. And so, you know, clearing of land for whatever reason is going to reduce the availability of food for insects, but also a shift towards more industrial food production also means that you know, bees have much less choice in the food that they can forage from. And combined with that means more exposure to pesticides, which are, you know, neurotoxins designed to kill insects, so highly likely to have an impact on either survival or the behaviour of these insects. Another area that's received a lot of research is the impact of novel and emerging pathogens on bee behaviour and mortality. So honeybees and more recently bumblebees get shipped all across the world for pollination. But unlike with other livestock, there isn't any kind of formal quarantine process for that. So that means any diseases that they might have get shipped with them. And so wherever they're being shipped to, the local wild bees there do not have kind of evolved strategies necessarily to cope with these pathogens. So that is also posing like an additional stressor to the bees. And of course, all of these different stressors are likely to be massively exacerbated by climate change. For bumblebees particularly, who are kind of a cold adapted insect, kind of unusual in that respect, they're going to be massively affected. And there's some really interesting evidence and studies, and it's not easy to do this work, but showing that there are interactive effects between these stressors as well. So, you know, having a poor diet makes you more susceptible to diseases if you're a bee. but also being exposed to pesticides has been shown to affect disease tolerance. There's some really interesting research showing changing climate and CO2 levels is affecting the nutritional quality of pollen, for example, available for bees to collect. But also changing temperatures might change your metabolism and change your nutritional needs in the first place. So all of these stresses are kind of interacting with each other. So I'm going to take a couple of these in turn now and talk about a little bit of research where behavioural approaches were used to kind of understand what the sublethal effects of these stressors and combinations of these stressors are having on pollinators. So more recently, I was invited to get involved in a kind of thought piece / modelling paper led by my PhD supervisor, Natalie Hempel, in collaboration with Sean Rands at Bristol University and Christina Batias, who I worked with as a postdoc as well, and who is a real expert on bee pathogens. And in this piece, which was really fun to be involved in, we kind of thought about what are the behavioural adaptations that bees already have that might help them to kind of adapt to novel pathogens or kind of just generally cope with pathogens in the environment. And there's some really interesting research by Pete Greystock, for example, and others that have shown that flowers that insects are visiting to collect food are real disease hotspots. And this is because when bees visit flowers, they like to poo, and many of the pathogens that they're exposed to are transmitted through fecal oral transmission. And often in kind of theoretical considerations or field studies where we think about pathogens in the environment and the risk posed to insects, it's quite common to think about the risk of bees being exposed to these pathogens as kind of being equal. So we can think about them as just little agents kind of bumbling around in the environment, bumping into flowers and picking up these pathogens. But of course, we know from kind of decades of behavioral research that that isn't how bees forage, that they are kind of actively making choices in the environment. And so there are a whole host of factors that could affect how likely they are to encounter these pathogens and what the actual risk is. And so we wrote this piece where we considered these options, and now we're doing some modelling work where we take an agent-based modelling approach to kind of think about different behavioural aspects and how that might affect the likelihood of becoming infected with these pathogens. So some of the things to consider is whether an insect is social or not. So if you live in a social colony, a lot of the transmission is likely to happen back at the nest, even if you're not a bee that's going out foraging. The foragers are going to be bringing those pathogens back. But of course, your size, either within a colony or between species, is also going to affect how far you fly and your foraging range. And again, how likely you are to encounter pathogens and the time scale over which you're going to encounter those. There's some really nice research that's shown that the morphology of those flowers, which is so varied, will really affect how likely it is that you that pathogens get deposited on the flowers, where the pathogens get deposited on the flowers, but also how quickly they're likely to break down. Many are not stable under UV exposure. So if a flower is quite complex and the bee is having to interact with that flower a lot to access the rewards, the likelihood of pathogen deposition might be higher, but also maybe where the pathogens are deposited could be inside the flower and less exposed to UV, for example. And of course, we know that there's a lot of learning and sensory cues that are involved in guiding those choices. And of course, bees don't necessarily visit every flower that's available to them. Many species exhibit constancy, so they would be ignoring some flowers in the patch, thereby kind of reducing their likelihood of coming into contact with them. But also, we know that they can pay attention to how recently a flower was visited, so they can detect these footprints and avoid landing on a flower. And typically that's been assumed to be an approach to avoid visiting empty flowers. But of course, it could also have a secondary benefit in that it avoids visiting flowers where a pathogen might have recently been deposited. And then the rewards themselves that the flowers are offering affect the choices that the bees make, but also they often contain secondary metabolites. And again, there's some really interesting research suggesting that bees might seek out certain metabolites that are effective against pathogens. So either reducing the infectivity of that pathogen or kind of self-medicating, if you like. And so really the crux of this paper is that we're making the case that if we consider all of these behavioural adaptations that bees employ to make them more effective and more efficient foragers, we might also see some kind of adaptations to avoiding infection with parasites as well. And all of these factors kind of combine to provide sort of individualised critical time windows when the bees might become susceptible to pathogen infection. So yeah, watch this space for some modelling work on that aims to kind of test out whether we're on the right track with what we're saying here. In terms of empirical work that I've been involved with on pathogens, so bees, like many other insects, are central place foragers, so that means that they need to be able to get back to their nest, of course. And so when they're foraging on flowers, it's no good if they then can't find their way back to give that food to their nestmates or their offspring, whether they're social or solitary. And so there's been a lot of interest in whether pathogens might have an impact on kind of how far the bees can fly, sort of depleting their energetic reserves, or also whether they might have cognitive impacts on how well they can kind of navigate either to the patch of flowers or back to the nest. And so straight after my PhD, I was involved in a project led by Rotherstead Research, which aimed to look at kind of the effect of pathogens on bees' orientation. So we know that individual foragers on their very first flight from the hive, or the first few, will perform these quite stereotypical orientation flights, which allows them to learn kind of the landscape features that tell them how to get back to the hive in the landscape. And so in this study, we were looking at what is the effect of two different pathogens. So one is Nosema serrani, which is a gut parasite that sits kind of in the lumen of the gut and almost competes with the bee for any reward, any nectar that's coming into the gut, or any food coming into the gut. And so it's thought to kind of affect the bee's energy reserves, and it's been shown to affect kind of feeding behaviours and sharing of food within the hive. And then a second pathogen that we looked at was deformed wing virus. And we were interested in this particularly because it concentrates in the tissue of the brain. So it's been suggested that it may affect more cognitive tasks, such as learning and the location of the hive, for example. And so I came into this project towards the end, so I didn't do this very hard work that Stefan Wolf, who was the postdoc on, did of inoculating these bees individually, newly emerged bees, hundreds of them, and marking them, and then releasing them into a colony, into a flight cage. And so I helped a bit with the tracking, but I did all of the molecular kind of analysis of the pathogens. So yeah, the bees were inoculated with pathogens, and then they were released into a colony that was within a massive flight cage, so that we could really control their experience, and we would know exactly when was their very first orientation flight. So when we were ready, they were allowed to leave the colony. We put a transponder on the back of the bee and then use the harmonic radar to kind of in real time track the orientation flights. And so in the right-hand panel, you can see kind of these flight paths of an individual, and hopefully you can see that they are doing this kind of fairly stereotypical looping behaviour away from the nest. And so we did this for several bees that had either been inoculated or just sham inoculated with sucrose. And then we plotted and analysed their flight path, looking at factors such as the length, the area, how far away they went from the hive, but also kind of those qualitative measures as well of the kind of pattern of the orientation flights as well. So looking at the sequence of straight lines to turning, to look at kind of that, give a qualitative measure that we could see whether that was impacted because the bees perhaps weren't performing these flights in a way that we might expect a normal non-infected bee to do. And then we used a combination of molecular and microscopy techniques to produce a pathogen load value for each individual bee that had been tracked. And so these are some example tracks from highly infected or not infected bees. And I just show these to illustrate, but obviously we also did some statistical analysis on the length and other parameters of the flight path. And so in the top left-hand panel, you can see what would be the control bees. So they have low pathogen loads of nosema and deformed wing virus, and they're performing these very stereotypical flights. But then the bees that had very high loads of nosema, so this gut parasite, the pattern or the kind of arrangement of their flight paths was similar. There was no kind of qualitative difference, but the overall length of these orientation flights was significantly shorter. So that does seem to suggest that those bees were not cognitively impaired, but they were simply running out of energy to perform as long orientation flights as the control bees. And this group also did a similar study where this time they took bees that knew the location of the hive, manually took them far away, released them and looked at navigation back to the colony. And again, they found that the bees were on the right heading direction, they're going the right way, but they just kept running out of energy on the way back. So again, nosema was really having a negative impact on the ability to perform these flight paths. Deformed wing virus bees, the kind of qualitative differences and the quantitative sort of lengths of the orientation flights were not affected. So this pathogen clearly was not having an impact on the ability to perform these orientation flights. But I think this is a nice demonstration of how pathogen infection can be affecting kind of these complex real world behaviors, which at an individual level, the bee might not be dying, but it could be behaving suboptimally, which could have, in a colony of thousands of workers, if they're all similarly affected, could be having effects on kind of the foraging workforce, for example. So that was a really fun project to be involved in. I then moved from Rothamsted to Sussex to work with Professor Dave Goulson and did a lot of work on pesticides, so looking at effects on bumblebees, but also solitary bees as well. And also a lot of work just trying to quantify what is the level of exposure that bees are regularly experiencing, because that was at the time quite a lot of criticism of kind of lab-based studies or behavioural studies was that we were giving them really high doses to kind of find certain effects. But actually what we found is that the doses that they are encountering, not just in the crop, but in wildflowers around crops was really, really high. And those studies were using concentrations that were kind of within the ranges that we were finding in the field. So neonics and other pesticides are neurotoxins. As I mentioned before, they're designed to kind of kill insects, but that also made them kind of an exciting challenge, should I say, for people who are interested in behaviour, because it's a way of bringing an applied element into your research, but still asking, what does happen if we mess up the synaptic connections in the brain? What are the knock-on effects on brain function and behaviour? So neonics work by disrupting the acetylcholine signaling system. So they sit in the receptors and they can't be broken down by acetylcholine esterase. So they're constantly sending the message to the receiving neuron, and at very high concentrations that causes kind of complete muscle spasm and to die. But obviously at sublethal levels, they could also be really messing up kind of learning or other kind of critical behaviors. And so there's been tons of work done on the effects of these particular pesticides, from the cellular level right up to kind of effects on colony level, but lots of behavioural work in between as well. And so I was involved in a study at Sussex where we were actually interested in looking at combined effects of stressors, so looking at the effects of pesticides and pathogens. And so we used a very popular behavioural assay called the proboscis extension response conditioning assay. which is used a lot with bees and other insects to a certain extent, and was really revolutionary in understanding kind of learning circuits in the insect brain. And really briefly, for anyone not familiar, it's a Pavlovian conditioning technique. So the bees are presented with sugar at the antennae, which causes the proboscis to reflexively extend. If you present a neutral odour that the bees are not familiar with, they don't extend the proboscis. They don't have any kind of learned association. But if you give them multiple pairings of the sugar reward at the antennae and this neutral odour, so the conditioned stimulus is the odour, then eventually, after say 3 pairings, it can be pretty quick for a healthy bee. You will get conditioned responses to the odour alone. So the bee comes to expect a reward and starts extending the proboscis. So this has been used a lot to look at kind of sublethal effects, particularly of pesticides, but here we wanted to look at the combined effects. So again, we inoculated bumblebees this time with Nosema cerani, and then they were placed into microcolonies that either did or didn't get a dose of neonics. So we had 4 treatments, control, 1 with pathogen only, 1 with pesticide only, and 1 that received both stressors. And we chose to use 1 part per billion clothianidin. This is a very conservative dose, although we were chronically exposing the bees. So that's why we wanted to use quite a low dose. But in the field, we were seeing concentrations of up to 80, 90, 100 parts per billion in some non-crop flowers. But around an average dose is 1 part per billion is well within the kind of levels that we're seeing in the crop. So just important to highlight that, I think. But actually what we found was no effect of either the pathogen or the parasite. And on the x-axis, you can see the number of trials, and on the y, the proportion of bees that are exhibiting this conditioned response to the odour alone. And then we also did a retention test, and actually you can see that if anything, the control bees have the lowest memory retention. And actually, we also found that the inoculation didn't work. So we checked that the spores were live with all of that due diligence, but for some reason, feeding these spores to bumblebees, which it is known to be infected to, didn't work in our case. So I just put it in there as a bit of a failure, but also, it's kind of interesting that we're directly feeding these spores to the bees and not seeing any infection. But of course, we keep the groups separately because they have been treated separately. And this really contrasts with other work that has used similar, maybe slightly higher doses of similar neonics, where people have found significant effects using this same paradigm. So again, I think it's important that we don't just extrapolate from one pesticide or from one bee species, and that we consider the doses and the species. And I guess that's kind of a problem with regulatory testing, is that often it's only done with one species or with a certain dose or with a single stressor alone. And we know that bees are likely to be exposed to kind of a whole suite of stressors. But of course, when you try to do interactive studies, then your sample size and it gets quite difficult, but important, I think, to look at the interactions. So finally, in the last 10 minutes or so, I just wanted to talk a bit about research that is ongoing in the lab. And as I say, we started the lab a few years ago, and we've been developing some new behavioural and physiological paradigms. And really, we're asking whether understanding the behaviour of kind of individual bees could be a technique to help improve pollination and kind of be a way of making agriculture more sustainable. So we know that our reliance around the world on insect pollinated crops is increasing. So we're eating more insect pollinated crops. And that pollination doesn't just affect yield, it can really affect the quality of the produce as well. So these strawberries are an example of ones that have had, the very dubious looking one has had very inadequate pollination, whereas to get the nice aesthetically pleasing strawberry, you need to have every one of the stigma on this strawberry flower needs to receive some pollen. And so only certain insects or certain number of visits from insects can provide that. But, aesthetics aside, we also know that pollination by insects improves the taste of food, but also the shelf life as well, which is kind of quite an important factor in making food production more sustainable, because a lot of food ends up being wasted and thrown away. And many crops around the world are exhibiting a pollination deficit, which means that they're not reaching the maximal yields that they could if they were being adequately visited by insects. And the stats from this one study conducted by a team at Reading University is kind of scary, that just for one variety of apple, the yield gap owed to insufficient pollination is worth about £5 million a year just in the UK. So, if you multiply that up by different varieties of apples, different crops, different countries, there is substantial kind of gaps to be closed there by improving the delivery of pollination services to crops. And so my work at the moment and the grant that allowed me to start my own research group is kind of based on this premise of, again, reminding ourselves that pollination is really dependent on individual choices of foraging bees. So thinking about these bees as agents that are making decisions and weighing up which flowers to visit. And excuse the massive oversimplification, but if we imagine a bee in a meadow where it has two options of flowers to visit, it might be constant and only visit the flowers of one variety. That's great for the plant because it means only the pollen from the same species is being deposited. Excellent. But depending on several factors, such as whether this is a social bee, how far this bee can forage, what is available in the landscape, the bee may be flitting between the different species. That's less good for the plant, for pollination, for the crop, for example, because you're getting heterospecific pollen deposited, which we know can disrupt pollination, seed sets, and that kind of thing. And basically everything that we know about what guides these foraging decisions and whether a bee would behave more like the first bee or the second bee is based on nectar rewards. So this has been studied for tens of years. We know that bees can adjust their behavior based on the volume of nectar, how sugary it is, how quickly it replenishes, and they're very economically minded. They're making these decisions. But we know really little about what guides pollen collection in bees. So this is for a couple of reasons. Pollen is quite nutritionally complex. It contains fats, proteins, lots of micronutrients. And nectar, we are also realizing, is not just sugar water. It's also more complex. It has these secondary metabolites. But it isn't until really quite recently that we've understood what bees might be trying to maximize. And it's also quite complex to collect. So it needs to be packed away into leg sacks. It requires quite complex behavior, and for quite a few bees or social bees, they might not be really ingesting it at the flower either, although there is some interesting research coming out now suggesting that maybe they are doing a bit more sampling at the flower than we thought. But until recently, it hasn't been quite clear kind of how much nutritional information do they get when they're foraging for it as compared to nectar, which is drunk directly at the flower. So the mechanisms of assessing the nutritional quality are quite clear. I think what's also important to consider is that most of our research and behavioural work particularly has been conducted on social bees, but social bees are kind of the not normal for bees or for pollinators. So most bees are solitary, and whether you're social or solitary is really likely to have an impact on kind of the rules that you use to kind of govern your foraging behavior and what you're trying to maximize. So if you're a social bee, you're bringing back resources into kind of a pooled shared load that gets kind of divided up between offspring. Whereas if you're a solitary bee, you're needing to bring back all of the food for your larvae. So you may choose to kind of exploit and explore habitats quite differently to a social bee, and the rules that you might use to kind of guide leaving a patch or something might be quite different compared to social bees. And so the project that we have at the moment is kind of approaching these questions from three different angles. So we're looking at the physiology of the bees. So we're asking whether they can taste nutritional compounds in the flowers. We're asking whether they use sort of non-nutritional cues to guide their behaviour? Are they factoring in how easy it is to access these rewards, to pack them? What happens if the nutritional cues and the behavioural cues required to access this reward are put into conflict? What would they be willing to kind of invest for a better or worse pollen reward? And then finally, we want to know what are the implications of those foraging decisions at an individual level for the deposition of pollen and for pollination of crops, for example. My postdoc, Sergio Rossoni, has a neuroscience background. So he is building on some of the work from the Wright Lab, for example, where they have been looking at sucrose perception and gustatory perception in bumblebees to do single centillum recordings on the mouth parts of bumblebees, and looking at dose response curves to amino acids in the first instance. The picture is complicated, and you can hear the full story if you're going to ICN this year. But the responses to different amino acids seem to vary quite a lot. Valine, the response is pretty reliable too, but we think that might actually be going through the sugar receptor anyway. But yeah, we're doing more work on this. We're looking at combinations of amino acids as well, because it's very unlikely that the bees experience just one amino acid in isolation. And we're also now looking at higher concentrations, but we wanted to get a kind of dose response curve right through the concentrations. So that work is ongoing. And then Sam, my PhD student, is doing work to look at the post-ingested cues. So myself and others have become more interested in recent years about this sampling at flowers, and I know Avery Russell has a new paper or a new preprint out looking at this kind of these kind of questions as well about, you know, what are the bees sampling at the flowers. So Sam has been collecting workers of different castes, bees of different castes from bumblebees, and looking at whether any pollen does end up in the gut and what is the fate of that pollen. Does it end up getting cracked or opened? What is the timeline of receiving any post-ingestive cues, and how might that shape decision making and foraging as well? And then Natasha, my postdoc, who is also a member of the Animal Behaviour Live organising and founding committee, she's taking the behavioural approach and looking at kind of how do the handling cues that are involved in extracting this reward, which I would argue are in some ways a bit more complex than for nectar, affect their foraging efficiency and the decision making. So we're using a behavioural setup with 3D printed flowers and kind of providing the bees with flowers of different orientations. So keeping the reward the same, but looking at changing the handling costs and looking at how that affects the decision making of bees that either have time to specialize or that are forced to switch a lot. And eventually what we'd like to do is put kind of the handling cues and the quality of the reward and the nutritional quality in conflict and look at how the bees respond and how that affects their choices. And then we're also doing some behavioural work combined with flow-through respirometry, where we're looking at how at quantifying the energetic cost of collecting pollen and other rewards, and trying to put a kind of value on the cost of engaging in this behaviour, which hasn't really been done before. Really excited about these experiments. They're quite challenging to get the bees to behave in such a small confined space and with airflow and things, but I think the results should be really interesting. And so what ultimately we would like to be able to do with this data is to, you know, imagine a farmer who is trying to design their agri-environment scheme. How do they keep the pollinator doing what they want them to do, which is to, you know, visit the crop and ideally stay in the crop and not deposit too much pollen from other flowers into the crop. Traditionally, they're advised to kind of plant a wildflower strip in the middle of the orchard. But could our research suggest that, okay, it's better maybe just to provide something that supplements what's already available? Or could it be better to kind of distribute these resources more evenly throughout the orchard, for example, particularly if you're relying on a species that is a solitary species that's trying to maximize kind of all of the different nutrients that it's trying to collect for the colony. So this is quite a long way off, but this is what we hope to be able to demonstrate at some point, and to take our kind of behavioural and physiological work and combine it and start to do some semi-field and field studies where we sort of start to test the predictions that we get from the lab. So in terms of take-home messages, I guess, I've talked a little bit about what some of the causes of declines are and how kind of studying behaviour might help us to understand it, but also potentially we could come up with some mitigating solutions, for example, to help bees meet their nutritional needs in our agricultural landscapes, but also to provide more sustainable food production solutions, where instead of relying more on increasing inputs into the system, we improve the pollination service to increase yields instead. So thanks very much for the invite, and thanks so much to my group and to students who've worked with us, and to all my collaborators and my funders. And yeah, I'm very interested to chat to people who would like to come and work with us. So do just get in touch if you're interested.
I guess that was a great talk. Really fascinating talk. I found it really interesting because I personally work with ants, and I also have like interest in social insects, for example. Uh, so while we wait for people to go through the talk and understanding and viewers to write the questions in the chat, I have a few questions of my own.
Mm-hmm.
Uh, so you talked about the sublethal effects of different pesticides or even like the nasoma. So how does a bee colony or like honeybee colony or any other bee colony essentially what is their colony defense or like colony response to that? Do they have any kind of immune response, or like do they have like a special way of tackling like an asthma infection or any kind of sublethal effects of any other pesticides that you are aware of?
That's a really good question. For the pesticides, I'm not so sure. Like, I don't think they have any way of, avoiding bringing back other other than the bee not making it back, I suppose. But I don't think they have any way of filtering it. And I believe it was Jerry Wright's group that showed that they can't perceive the neonics, for example, in solution. So they actually, and I think there was even work showing that they actually quite like to drink the neonic solution because they get a bit of a buzz from it in low doses. So they definitely don't seem to have much defense against the pesticides. But for the pathogens, I think it depends whether they co-evolved with them. So Nosema cerani comes from the Asian honeybee, Apis cerana. So I don't know so much about their biology, but I imagine they have more defenses because they will have kind of co-evolved with this pathogen. But I believe there is some work showing that those bees that are infected would be less inclined to engage in trophallaxis, so food sharing, the ones that are infected. And I need to check that. But also, I think this self-medication work is really interesting. So for Crithidia, which is a protozoan, there's evidence that heather nectar can kind of affect the like cilia on that protozoa. So they do have some defense mechanisms, I guess. Yeah.
Okay. We have a few questions from a chat member, a viewer. So Valen98 is saying, thank you for the talk. I had two questions. The first question is, how would you address the complexity of olfactory cues associated with floral cues in general?
Like how to disentangle them, I guess, do they mean?
Yes, I
Yeah.
think so.
So I think that's a really interesting question. So there's work showing that the bees can definitely distinguish between the cues that are produced by the petals, for example, the olfactory cues, and the pollen. So bees can definitely recognize the pollen specific cues, But I'm sure when it comes to kind of making a foraging decision that these two cues interact, and so I think it is difficult, and what we are doing in the lab is very oversimplified as a kind of starting point to look at specifically for this reward, what is driving the choices. But it is complicated because I didn't mention that as another factor of pollen is that it has a strong olfactory component as well, which could be a cue that the bees rely on to determine the quality of it as well. We just have so little work on that so far. So yeah.
And they have another question. Have you considered any neuroimaging studies in your current work? Any neuroimaging studies?
No, not yet, but I think that could be an interesting direction for the future. So I'm not a neuroscientist, so I'm really dipping my toe in with the extracellular recordings. But and there is some work in honeybees, but yeah, that could be an interesting technique to bring in. Sure.
Yeah, I have. While we wait for more questions, I have another question. So does it this Nasoma and like the other pesticides, does it have like differential impact on like different caste of work or like different caste of honeybees? For example, I know in some species of ants, the queens are better at like having like neurotoxicoids, insecticides, handling insecticides compared to like workers. Does it have like a different level of impact on different caste? And can they recognize affected individuals? Can they, essentially like help them more or like do more, take care of them more, the infected individuals?
So the first question, I think that's really interesting, and I can't think of any work that has looked at. I mean, the work on solitary bees, in a way, you're looking at the reproductives, right? Because they're all reproductive, and so people have looked at kind of effects on reproduction, number of offspring. But for the social bees, and there might be, but I actually can't think of any papers where they've looked at specifically the effects on males or on. Well, maybe people have looked at not behavioural effects, but effects on kind of sperm quality and that kind of thing. But yeah, I think it would be really interesting because for bumblebees, the queen stage is a really it's a really vulnerable stage, right? Because they have to be able to overwinter, they have to be able to forage effectively because they're essentially like a solitary bee at the start of the year until they rear their workers. So maybe there is some work, but it is quite difficult to work with queens because, well, at least in my experience, they're not the easiest to rear wild queens in the lab. But yeah, I think that's such an important question to think about the effects on not only the less studied species but the less studied castes as well. And yeah, your second question, I'm not sure if they can recognize infected individuals in the species that I've worked on, and help them, probably not, maybe remove them. It's more likely in the social insects, I feel like.
We have another question from another viewer. So a question from Catherine. Thank you for your talk. I was wondering if there is any work showing a link between climate changes and the stressors, like a virus surviving longer on the flowers, for example.
That's a good question. I know people have looked at like the time scale of how quickly they degrade and on different flower morphologies, but I don't know if anyone's looked at higher UV or ozone levels or that kind of thing. But I think there is quite a lot. We have a project now where we'll be looking at the impacts of warming temperatures on nutritional needs, and then how that has a knock-on effect on foraging behaviour. So, and I have a PhD student who just looked at the effects of kind of short-term heat waves again on kind of behaviour in the nest, and you find that it does have some impact on kind of like other maintenance behaviors in the nest because the bees are having to divert
Mm.
time to wing fanning and trying to cool the nest and that kind of thing. So I'm most familiar with the work on the impacts of changing climate on nutritional needs and foraging and on the plants themselves as well. Yeah, great question.
We have another question in the chat from Abby George. Great talk. I'm wondering if there are any students that look at the effect of pesticides on both the individual and colony scales. I feel that most studies don't bridge the gap between these two.
Hmm. That's really interesting. Yeah, I think that could be true. I mean, there are some cool studies from like I think it's Nigel Rain and Richard Gill uh, looking at kind of looking at whole colony level, but then also pairing that with RFID to look at the individual level how much foraging is happening. Yeah, that's a really good point. Like it's often done in separate studies where we look at the whole colony level effect and then the learning effect. I guess because it's pretty labor intensive. So and if you're doing things at the individual level, then you're taking in like for the social insects, you're kind of taking them out of the colony. But yeah, nothing is springing to mind, but that's a really good question and maybe a cool direction for future research to consider within the same study the individual and colony level effects.
Uh, Catherine mentioned, thank you for your answer. Very interesting work.
Oh, thank you.
Uh, Also, I think we are almost at the end of the time. I would like to talk about one more thing. We are looking at your profile and we saw like you have received a lot of recognition for your work, but you have also done a lot of public outreach work. Would you like to share some of your favorite public outreach work or some of the works so we are also aware about it?
Oh, thank you. Yeah, I think when you work on bees, as I say, people really like bees, so it's quite easy. And but I also think people are really fascinated by animal behavior as well, right? That's what a lot of our wildlife documentaries are really centered on behavior. But I really like working with kids. I think they always ask the most fun questions. So I've done a lot of going to schools and kind of talking about the importance of pollinators for food production, but also some of the weird and wonderful behaviors that they get up to. But the kids always ask really fun questions like, one of them asked me if bees ever get any time off. Which I thought was a really good question and actually made me think. And you can still bring in the science because there is some really interesting research about division of labor in the hive and that there are, lazy ants or lazy bees that
Yep.
only get activated when colony demands are high and things. So, yeah, I really like doing work with schools, especially because I think that's where we can have really big impact at kind of um, informing and engaging like the younger generations in science and in our research.
Um, I think we are at the end of our time. It
Oh,
was really, I would like to thank you again for joining us today. It was a really great talk and also a really interesting discussion. And we also got to know more about how researchers are working to save the bees, as well as how you can integrate both behavior as well as uh, direct work from it. Uh, so if you guys have the for the viewers in the chat, if you guys have any question, please make sure to email Dr. Nicholls. She showed her email. Also, you can contact her on X and send her any question that you might have. Uh, we are almost at the end of our talk for today. Before we end, I would like to remind you guys that uh, our flagship event of Animal Behavior Live, the Animal Behavior Live conference is going to be happening on 4th and 5th of November. We have a we have a diverse list of plenaries. This is our fifth annual conference on 4th and 5th of November. If you want to join the conference, for some reason my screen is not working. Okay, if you want to join the conference, please make sure to sign up through this sheet. Okay. Okay, yeah. So if you want to join the conference, please make sure to sign up through this sheet. You can see the QR code and make sure to submit your abstracts by August 31st and register online. This is a free conference. We always prefer for inclusivity and sustainability and accessibility. There's going to be a lot of talks, posters, meet the PI session and workshop, more information on the website. So if you have any questions, please let us know. Otherwise, again, I would like to thank uh, Dr. Nicholls for giving us an amazing talk. And thank you again for everyone to attend and then participating in the chat. And for our viewers, make sure to keep an eye on our social media account for more information on the upcoming seminars, the conference, and other events. And we'll see you guys next time.